completely not the same author. So he started with Sudoku, and our work is related. So naturally, I'm going to focus, but the connection to his work. I'm going to try to give the same field. So this is joint work with Nonamir and Untitled. All right. So and how to identify rare alleles here, using which we call compressed sequencing. So again, two works that motivated our DNA by Yaniv Erlich and colleagues, and their application was finding defected microRNAs in large set of many wells. And another work by Bravo and Peer, where their goal was to identify carriers, in fact, a single carrier of rare mutation. The thing about those works, they use a smart idea called combinatorial. Those the nature of the problem and the exact pull strategy was different. So the pull strategy based on what's called the Chinese remainder theorem, and in Prabhu they used correction cause pull. Right, so now we'll go to our work. So first of all, what problem? So you got a population, a bunch of people, and you're interested in finding a mutation and the carrier of a specific mutation, for example, a SNP. So you go from here, from the set of individuals to the genotypes, and specifically you want to find the carrier of some rare mutation. So here marked in red. So the wild or reference genotype is A, and humans are diploid, so it's A, and the carriers are AB. And specific characteristics of the problem are, first we want to screen large populations. We're talking about hundreds or thousands of people. Second, and very important, the alleles you're after are rare. So most people don't carry the mutation. Only a few of them do. Third, you're looking for predefined genomic regions. For example, there is some gene protein or some other regions that came up in a gene, for example. You're not talking about full genome sequencing here. To focus on some specific regions. All right, so that's a problem. So the standard approach is simply you coordinate from each. Then there are new experimental protocols to target the regions you're after. So for example, if it's just, you can use the PCR as an amplify. If there are more regions, called hybrid solutions that enable you to enrich for the DNA regions you're interested in. So you sort of get rid of the genome, and you just sequence using next sequencing. So after you target your regions, you perform one for each individual, so one length, for example, and the carrier. So this should. So you see that by definition, if is rare, it means that in order to, for example, if the frequency is 1 or 1,000 of individuals, and probably hundreds or thousands of people, and you waste regions. So, for example, in today's tens of millions of reads, you sort of waste and give any extra information. By using multiple cell, and you can pull identifying each individual. So sample one line. So that's a nice solution. It doesn't scale to a very large different records. we offer a computation solution. And again, it's a form of pulling them. It's very similar to what was done in the Sudoku. So instead of performing a run per individual, you create different pools. You see combinatorially. So every pool individuals, and every individual participates in pools. So it creates pools, and pools runs their individuals separately. And okay, so that's the sequencing step. Now you have the 6 reads from each pool, and now there's a reconstruction step that will take you through the sequences of each pool back to the individual and find the carriers. The strategy is where you have your pools, so the number of pools you do are the number of individuals. This means that it costs less in sample preparation, and you still get the correct. Right? Clear so far? Okay, so why this problem? So I'll just give 2 applications. One is a carrier stream. So we all know about the rare recessive diseases. So that's a disease where you have some we have some SNP that is SNP, or it could be also a small insertion deletion, which is causal for a certain Mendelian disease. And let's say it's recessive. So if both copies are wild type, you're healthy. If one copy is a disease-causing variant, you're healthy. Only if you got both copies, then you're sick. So this application, we are trying to identify areas of known mutations. So we know what mutation, what it is, etc., and we're trying to find the carriers. This is our law of inheritance. So we know that if both parents carries a mutation, there is a 1 to 4 chance that the offspring will alter. So we want to do this before mating, for example. Then it tells parents, look, there's a chance that they are. And as this example of many of these diseases, and the important thing is you see that the carrier rate is pretty low. I mean, it's not absolutely low. I mean, it's still on the order of maybe a few 0.5%. But again, the disease is only in the population. So if you screen a population of 100,000 of individuals, most of them not carry the allele. All right, and now when I talk about specific mutation, just to illustrate what I'm talking about, so we have the reference human genome, and then you can either look at the specific SNP, or let's say a deletion, and we mark the wild type by A and the mutant or disease-causing allele by B. And again, our goal is to find individuals with AB, or also mutant homozygous BB. So that's another application in the GWAS. So genome-wise association, you suppose you screen for certain diseases and have controls. And then you look at many places, profile all individuals for these variants. For this, for example, one variant, so A, B, etc. And you look at markers that have differences between cases and containing more A's in controls than in cases, and this locus is in about hundreds of thousands or millions of images found. But what was found was hanging fruit. So I'll just explain this slide. So first, look at free population. So here we're going to count to rare SNPs, and you can look at the effect size. So how frequent between cases and all. So the lower the effect, it's a find variant. So on variants for using lineage analysis. What you in today is about this front, found those low in the field move further. So to find those rare variants, that are rare, we need again to project populations. For example, we just look at regions interested in. You want a large population with lower frequency, and popular, which has been some regions of interest and just research. In both applications, it doesn't matter if the mutation you're after is known or you're trying to find new mutations in some region. The mathematical problem is the same. You have a set of regions and you want to find the mutations. All right, so what is our solution? It's called compressed sensing based group testing. So we have the population and we have two carriers. So we take one pool. Okay, we pull them, and then we measure the frequency of the B allele in the one here, both in the pool. So that's 1 + 1, and divide by 5, which is the number of people, times 2, because we have 2 alleles for each individual. So we have a measurement which is the frequency of B alleles in the pool rather than for a certain individual. Then you perform another pool. Here's the frequency is 0 because we didn't pick any of the carriers. Then another pool. Here's the frequency is 110. And then again, we're trying to read from this frequency the identity of the carriers. So then I'll tell you how is it step done. So the first step is the measurement of next generation sequence, and the reconstruction is done by a technique for statistics. All right, so now we'll just formulate the problem as a compressed sensing problem. So what we're after is this vector of unknown. So each entry corresponds to an individual, so it can be AA, AB, or BB. And we denote it by a vector of integers where 0 means AA, 1 means AB, and 2, which is not here, means B. So basically, the entry just tells you how many B alleles you have for each individual. So this is the unknown vector we're trying to reconstruct. Now, what is a measurement? A measurement or a pool can be represented again by a row vector, where you put 1 whenever the particular individual participates in the pool. So, for example, here, 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 and here. And you normalize by the total size of the pool. And then you have a measurement. So yi is the measurement size pool. It's simply a dot product of the pooling vector and the unknown genotype vector, right? Because whenever you have a B here and it participates in the pool, it contributes to this frequency. So it's simply a dot product. All right, so we have y. We know the pooling vector, and we're trying to reconstruct x. So that's a very simple problem. It's a problem, just a bunch of linear equations. And that's also the standard compressed sensing problem. The only problem here is that we have far fewer equations than unknowns. So in general, when you have less equations than unknowns and you have a linear system, we all know from linear algebra that you cannot really solve this problem uniquely, right? There are multiple solutions x. However, in this case, we have more information. The crucial information is that x is par. So now this is the L0 norm of x, which means basically x that are not 0. And we know that it's much smaller than the in genotype text, an entry here being 0, and 2 entries which are not 0 means mutant. The solution is possible. And there is a very nice tell that if the solution is sparse and the matrix pulling certain properties, then it's two things. First of all, uniquely, which is very nice. You have much fewer equations. Second, not less important because we're talking about algorithms that can do that. And I want one of key idea is to move from the L to L is the convex optimization problem. So as long as we know this few pools information, we can save a few pools or a few equations in this context. And types. All right. So now we in practice how to measure these frequencies. So it's real lines to the genome interested in specific location. And the difference is simply look at the specific location, just look at all the reads and count how many reads allele. We have to see the blue you see. In this example, we're talking about one pool, and we sequence it to a pool this particular location. The frequency, because we've seen reads aligned here, and only one of them was T. So in theory, everything is nice. The measurement is exactly the dot product of the pooling vector and the unknown VG type. In practice, as you might notice, there are many errors. For example, we don't observe the exact frequency of the BLAs in the pool. We just observe a band for reads. So how we model that? We model that by a binomial distribution. So we have the unknown frequency yi, and we observe, let's say, 16 reads here. So the number of reads which we really observe is modeled by a binomial distribution. So in practice, Zi, the number observed, will only approximate Yi, the true frequency. So that's one source of noise. There are many other sources of noise. For example, the coverage will not be unique. So if you amplify different regions in the genome, and you actually do this experiment, you can get different coverage for different regions, which means that for the regions with high coverage, it will be more accurate. For regions with less coverage, it will be less. So we also model that, coverage. Also other sort of technical errors, for example, real machines and DNA function errors. So it's very hard when you create a pool, keep the amount of DNA from individual in. There are fluctuations in functional amplification efficiency. So we model all these of errors, and eventually, instead of this linear equation, we have a more complicated problem, which holds the term. So we need to find the solution. So we're here with trying to find which are. We also keep the error low. So this is the optimization problem. And how do we so we measure how by how much save in terms of cost. So here's the number of pools you do for lane. Here's the population size. So how many individuals can we track in this line in most of our performance to be very restrictive. So it requires instruction that sequencing machines has errors trying to conserve it. So in the black line, the performance of the naive, namely you use one line per. So for example, with spiral lines, you construct 500 individuals. And well, the performance is best in our more than 10,000 people using only 200 lines. So that's the first measure. So it's only a very large amount of resources. So we may be dividing this number of ways. So in this example, you say up to 50 cost. So that's the first one. The second, more a little, the more safe. But when you go to, we still see them a little frequent verge to the performance. The number of people you're going to profile, so the population size. Here, but we can profile 10,000 people with the length again, length correspond to a certain pool. That's yeah. The exact performance depends on how many regions you're trying to do multi, right? So this is for a specific case. If you're trying to get, let's say, I don't know, 8 different regions, this number will change. Right. Next year of machines, the throughput will go, get more saving. Yeah. So all this coverage considerations as region size you target, the result, and the exact result depends. So that's okay. Easily varying all these parameters. There is a description by Yaniv Erlich, which has a software package which, if you tell them how many reads, how many regions you want, targets, etc., and so that's for people trying to do this kind of pooling. And okay, so approach, then I've shown you the other paper try to compare the different approaches. So pooling is common. All of these approaches, how you design the pools and how many algorithms are differ, and it's clear what is the best approach or between approaches. They're not that big. In simulations, we're trying to validate our approach with using real data. Unfortunately, there is no such real data available of tens or hundreds of pools for a large population. We have two data sets that provide partial validations. So one is the real pooling experiment, but in which only one pool was done. And the other one is a large-scale experiment from the 1,000 genomes data in which each individual was sequenced by a single lane. So that data set is from a paper by Out et al. There are 88 people in one pool, they one region, and sequenced a lane. And they also are using Sanger, that also gives you the ground truth. And on 25 SNPs, we only care about rare SNPs, so we need to but we need to create any pools, so they have only one pool, and we have a procedure for resampling and creating many in-silico pools. All right. So variation and technical issues that come into making pools and next generation sequencing come into this read. So this is our simulation represents this technical. Then again, we apply algorithms. Sorry. And on the most part, in red we have our simulation-based model. In black we have the resampler. So in this case, we have perfect match, performance of the data. But when we went in which frequency in the pool, we don't know why. You should try to the pool match the frequency in the population. Predict pretty well. And again, another data set. This is the previous data set. We had only one pool and one region, so it gives us statistics. This is, as far as I know, the most large-scale experiment. 1,000 genome data, and we looked at 364 individuals sequenced by the same platform, Illumina, as individuals are using process we discussed them. And there are dozens which are sequenced. We look only at identified SNPs which are rare again. We have 600 SNPs. Again, we create a silico pool. Here's the silico bootstrap population details are a bit different because the actual data is just one line to read, and so we had to do sort of pooling ourselves. Again, compute. And the performance predicted statistically with actual performance, which is in are just two different ways. Reports or validate, and is actually performing a large-scale experiment and shows that it works in practice, and you can find new mutations at low cost. So let's load. So we have a generic approach. You combine sequencing and compressed sensing. It finds both the heterozygote and homozygote. You can find SNPs, you can find indels, and it saves you a lot of resources compared to an approach. And I think I talked briefly about barcoding. So if you do barcoding, let's say you can barcode 20 samples per line, you can still find an easy approach with barcoding and save even more. So in that case, there's two solutions that are synergetic. And we have a script, we have new, and I just want to thank my collaborators, again, Noam Shental and Amnon Amir from Israel, and Yaniv Erlich, who is here today, but we now collaborate with him as well. And thank you. I'll be happy to
talk. Thanks for a great talk. Do we have any questions for the speaker?
Are you planning on looking, because the main problem with rare alleles or build rare alleles is to do a genotypic phenotypic coordination would be, and first step towards that would be to multiple rare alleles. So that sort of follows the question I asked. Would you be trying to look and see how your methods perform in finding multiple rare alleles, overhead savings you have in that respect?
Okay, so the question is, can we also use it to find multiple alleles?
Correct.
Right. So as I understand, in your talk, you have, let's say, 5 or 10 regions. In each one of them, you know that there is a allele.
Different regions, I mean.
Yeah, different
regions.
Yeah.
I want to see a combination.
All right. Yes, so it does that. I mean, it doesn't matter if it's one region or one region. You're right that the more regions you have, the less thing in performance you have, because this means that the coverage for an individual region will go down, right? So this is one parameter of our software. So you can play. I can't tell you off the top of my head the exact performance. Yes, we have a parameter. That, also in the manuscript we have this in the length of. We didn't look at multiple regions, but that's of the which is about. So the longer the region you're trying to find, reads you have per loss. So yeah, the top reads in length, which again is going up very rapidly, and then another parameter is the size of the regions you're trying to coverage, and our algorithms take both. Right.
So, I have two questions. So my first question for you is, in target sequencing right now, solution capture genome-wide for access, there are a finite number of molecules to do the targeted action. And one of your assumptions is that for individual DNA, you're going to be able to In fact, the current technology, we're barely able to sequence deeply enough given one capture kit for a given individual. So part of the limiting step is actually the capture technology. Have you all thought about
Yeah, I'm certainly not an expert in the experimental protocol, but I do experiments in the Broad Institute, and they are doing this hybrid selection. There is this that if you pull too many people, then many of them will not go into library. So I can't tell you on top of my head like what how to put before we actually do the experiments. They've been doing experiments with about 3 people per pool, and it works fine. And they say going on that they're sort of scared. Yeah, they say exactly because of. So what we're doing now is actually simulate. So let's say you have this pooling matrix, and it can be 1,000 even more, but we're trying to simulate sparse pools. So in each pool, we put in like 50 or 100 pages. And you see that the performance does not rate rapid. So you can make your pools more and more sparse until a certain point. You also get the same performance. Only when the pools are very, very sparse, let's say 10 people in, then our performance goes down.
Another related question is, ISCB peers group has taken individuals and arraying them out, one set of individuals and individuals, every column and row of get into a given pool, so that every individual winds up appearing in at least one column or one row's worth of pools. So you can think of a nice pool, say 1,000 individuals, and every individual appears in several of the pools. Have you all thought about combining your strategy with that array pooling strategy?
No, so you're talking about ISCB? Yeah.
Yes.
Okay. Yes. Yeah. So they have a very fully designed pooling strategy based on error correction codes. You're right, with this matrix. So as far as I understood, their method performs better than that, but it's only able to detect one carrier. Here we are, even if there are, let's say, 5 carriers out of 1,000, we can find them. So the nice thing about this is the theory tells you exactly if you do pooling, so you just put each per individual at random, then your performance is almost optimal. So in that case, we just random. It is true that if you carefully design or you might improve a little bit, but the market, according to theory, is not large, right? So having sort of optimizing the pooling is.
Great. Yeah, it could be interesting to see how it would be. Yeah. Are there any other questions for the speaker?
Yeah, I have a comment which probably isn't practical, but it reminds me of a problem where you have 2 stacks of coins, 10 each, and one stack is heavier than the others. And it's an old chestnut that the best way to do it, if you have a on a scale, is simply take 1 from the first pile, 2 from the second, 3 from the third, and so forth. You can identify it immediately. I just wondered, all the strategies in encoding or the coding all seem to be making sure you know or equal numbers from each individual. Would it helpful at all to have different amounts from each individual?
Uneven pooling distribution.
Yes. So if I understood the remark, so it's very important that it takes the same amount of DNA from each individual.
Well, the way you're structuring your code, yeah, but as the problem I mentioned indicates, you do a lot better. And the ideal is if you do that, wouldn't make any difference in your case if you took uneven pooling.
The suggestion is to use pooling, but take different amount of DNA from each individual.
Has anyone looked at that? Or
Yeah, I certainly haven't thought about that, but it might be worth thinking about. Thanks.
Okay, great. Well, thanks for a great study, and this is an important topic. I'm sure we're going to be hearing more from you as time. All right, so